All right, welcome everybody to the second part of our Chapter 1 discussion. So this is the second video to our first chapter, and this will be the last video for the chapter. So only two videos for this first chapter. So far, we've discussed kind of an intro to biology. So like, what is biology? What does biology study? How does that relate to human biology? And then kind of how do we and so what are the characteristics of living organisms? And basically, how do we classify living organisms? Now, what we're going to finish up with on this chapter is briefly discussing the level of biological organization that we see in organisms. And um, this is kind of related to the level that we look at this organization from, say, the molecular level all the way up to kind of the broad scale world view or biosphere view of ecological or biological organization. And so when we look at the smallest level of organization, so the smallest unit of matter that comprises living organisms. So, for example, humans, at our smallest level, we have our atoms, right? And so these atoms could be maybe oxygen atoms or carbon atoms. And then you can take multiple atoms, right, and put those together, and you can make molecules, right? So, like an oxygen molecule could be O2, water molecule will be H2O, methane, CH4, so on and so forth. But then we can take these larger molecules and look at how they are put together to make up macromolecules. And we'll talk much more about macromolecules whenever we talk about organic compounds and whenever we talk about cells, because we'll talk about how molecules like phospholipids come together to make the plasma membrane that surrounds the cell and keeps everything inside that cellular environment. Then whenever you have those macromolecules and you kind of put them all together in various forms and functions, you get a working cell. And now the cell is the smallest unit of a living organism. It's the smallest unit of life. Because you can have a single-celled organism that is alive. Everything lower in that biological hierarchy, so the atoms, the molecules, and the macromolecules, they're not living, but they are very important in making up a living organism, so that cellular component. But now whenever you have your multiple cells put together for the same structure and same function, that's what tissues are. So tissues are just kind of a grouping or an aggregate of cells that are serving a very similar purpose, right? So like muscle tissue or connective tissue, nervous tissue, etc. And then whenever you have multiple tissue layers put together, they can be combined to make an organ. And an organ has several tissue types that are carrying out a specific function, a common function. And then when you have multiple organs carrying out a common function, right, they make up an organ system. So, for example, the circulatory system is there to transport blood to the organs and the various tissues and cells that are around the body. So all of the organs that are part of the circulatory system are their purpose, their function is to move the blood around the body so that oxygen and carbon dioxide can be dropped off at various places. Now, once you have several different organ systems put together, so for example, the human, we have around 11 different organ systems, that together makes up an organism, so a single individual, so one human being. Then the multiple organisms will make up a population, and populations can be broadly defined based on how you want to classify it, but it's basically looking at all of the organisms that live in the same area, so in a single area. And so we could have like the population of Merced College, or the population of the city of Merced, or the population of Central California, or all of California, or maybe the population of the US, right? And so we can define populations differently depending on the scale that we're looking at. And so again, a population is organisms of the same species that are living together in the same area. So for example, all humans living in the same area. But now we have multiple populations that are going to live in the same area. So multiple populations of species. So for example, you'll have humans living on and going and visiting Merced campus. But on Merced campus, we also have trees. We also have squirrels. We have birds. And all of those organisms have their population that is on Merced campus. And those interacting populations make up a community. And so it's several populations of different species that are living together in the same area, and that's what we define as a community. Now, for an ecosystem, it's all of the organisms, so the community in an area, plus the non-living factors of that area. So that's the non-living matter, the non-living energy. So it's what we call the abiotic factors. Right? So let me kind of just write this out. So that would be abiotic. Right? So abiotic is non-living. So I'm going to put NL for non-living. And then the biotic are the living factors. So we'll put an L for living. So the biotic is the community, right? And so we go from a community to an ecosystem by adding in those abiotic factors. And some examples of abiotic factors are like soil, climate, rock composition, nutrient composition, right? And so all of those non-living factors interacting with those living communities make up an ecosystem. And then finally, the most encompassing level of this hierarchy is the biosphere. And the biosphere is all of the ecosystems combined that make up Earth. And these ecosystems, the biosphere, is the living portion of Earth, right? That contains all of these ecosystems. Okay. So here we have a table that kind of defines all of those terms. And then we have some issues and controversies that are related to those terms. You don't really have to worry about issues and controversies, but they're here for you to basically as a FYI for you if you're interested to look more into. What I really want you to focus on are the definitions and what we mean by each of these levels of organization. And then I want you to also be able to tell me which one is more specific and which one is more broad. So which one encompasses more things. So biosphere encompasses much more than an atom. So an atom is really specific, right? An atom is the smallest unit of matter. And you also want to know that a cell is the smallest unit of life. So, right, that is the lowest level of this hierarchy that we classify as living. So you want to make sure you're familiar with this level of organization and the definitions associated with them. And then here we just have a figure that's showing kind of that hierarchy as you go from more specific to more broad, right? So from atom to molecule to cell to tissue, organ system, organisms, populations, communities, ecosystems, and then the biosphere. Okay? And then also we can add here, because we will talk about them, we can add macro molecules. All right, sorry about chicken scratch, but that's the big thing is the macromolecules, right? So those are the molecules we'll talk about in the next chapter. All right, so what we're going to finish up with for this chapter, and basically the last half of the chapter, is discussing science and what exactly is the process of science and how we can make choices and decisions about the information we're given on science and the data that scientists will provide and give to the public. So basically, science is just the study of the natural world, right? We've already mentioned this, and we said that biology is a part of science, and biology is specifically looking at the study of living organisms. And specifically for this course, we're going to focus on humans as those living organisms. And science can be broken down more specifically into two components. One is knowledge about the natural world, so basically what we know from the process of science. And that second part is the process that we use to generate the knowledge that we know, right? This process is what we call the scientific method. And so you're probably familiar with the scientific method, maybe at least a little bit from, say, like high school science or, any other science courses that you may have taken before, or you may have at least heard of it being used, by somebody else throughout your life. And what we're going to do now is we're going to break down what exactly the scientific method is and the different steps that are important for the scientific method and that process of science that allows us to gain knowledge through the scientific process. So the scientific method is basically a process that we use to test ideas that we generate by making observations on things that happen in the natural world, right? And so the first step in the scientific method is that we have to observe something happening. And when we observe some phenomenon or action or behavior, we then can make generalizations about that behavior, right? So one example that I always use in a lot of my classes is like a flower example. So let's say in your backyard, you have a shrub and you have multiple shrubs, right? Of these shrubs, one produces purple flowers and one produces white flowers. And let's say you notice that bees will only go to the purple flowers and they never go to the white flowers. And you notice this day after day. Bees are always on the purple flowers, but never on the white flowers. So that's an observation that you've made. And you can generally say bees only go to purple flowers instead of white flowers. Now you can take that generalization and you can develop a hypothesis and you can actually test whether or not your observation is true. And so you can formulate a hypothesis, which basically says that bees only visit purple flowers, or bees visit purple flower shrubs more often than they visit white flowered shrubs. Okay. Now we can test that prediction, and a testable and predictions have to be testable, just like hypotheses. Actually should be this way, right? So hypotheses have to be testable, and you want to generate them in a way that they can be tested, right? So we can actually test whether or not purple flowers get more bees on average than white flowers, right? And we can set up an experiment to test that, right? And we can make a prediction that purple flowers over, say, a month's period of time will get more bees than white flowers. And we can experiment, we can make observations. So sometimes you have to manipulate the environment or manipulate what you're looking at in order to experiment on it to get an answer, or you can just make observations. So in our case, we can set up a video camera and we can watch that video back and we can count how many bees go to purple flowers and how many bees go to white flowers over a set period of time. And then we can generate a data table and we can look at our data and we can say, on average, do the shrubs get more bees based off of the different color of their flowers. And we can look at our data, and then if we see that our data holds true, right? So we see, yes, bees do visit the purple flowers more often than the white flowers. We can modify our hypothesis and maybe generate another question, and it could say, okay, now we can look at, why do bees visit purple flowers more often than white flowers? Is it that the purple flowers generate a specific smell? Can bees only see in the color spectrum, the ultraviolet spectrum, or the electromagnetic radiation, a certain value in those wavelengths that only the purple flowers give off versus the white flowers? And so you could generate other questions from that and continue to refine our understanding on why bees only go to purple flowers versus white flowers. Or if our observations are false, right? If our hypothesis is false and bees actually, maybe they go to white flowers more often and we just saw an anomaly over that one day, or maybe they go to them the same amount, then we can modify our hypothesis, retest, and continue from there. And so overall, the scientific process is actually this cyclical process that goes through reiterations over and over again, and is constantly refining the observations and the questions that we're making. So we, again, generally make an observation, we make a generalization, we formulate a hypothesis, we make a testable prediction, we experiment or observe to gain our data, and then we look at whether or not our hypothesis was proven to be false or not. And then we modify our hypothesis as necessary, and we continue to go through this cycle of the scientific method to increase our understanding and to increase the confidence that we have in our hypothesis and our predictions. And one thing I want to note is that hypotheses, not only do they have to be testable, but they also have to be able to be falsified. We are trying to prove a hypothesis false. If we can never say that our hypothesis is false, then we assume it to be true. So we're never really proving something to be true. If we can't falsify it, then that indirectly says that it must be the case, right? And so through our observations and generalizations, we're using the process of inductive reasoning to make these generalizations. And so we're just looking at observations and we're making that generalization through this inductive reasoning process. And so another example would be, okay, we make the observation that every winter in the past was always colder than the summer. So we can generalize that winter will always be colder than summer, right? So that's a generalization you can make based off of that observation. So another one would be, every giraffe I have ever seen has a long neck, so all giraffes will have long necks, right? And then once you make that generalization, you then make your hypothesis, which again is that statement, a tentative statement about the natural world. And based on your hypothesis, you're going to make a prediction, okay? And the hypothesis that you generate should be tested under several different conditions, right? You don't want to just test it once and then be like, okay, we couldn't falsify it, so that's the truth, and leave it at that. You want to rigorously test your hypothesis under different conditions, under different variables, to make sure that the generalization and the observations that you're making are accurate and haven't been able to have been falsified. You also want to make your hypotheses into predictions that are testable. And so your predictions are based on your hypotheses, and we use deductive reasoning to generate those testable predictions. And generally, your predictions are going to be in the form of if-then statements. So basically, if I do this, then this should happen, right? So like, when the batteries die in your remote, you can make a prediction. If I replace the batteries, then the remote will work, right? Most of the time, that's the case. You replace your batteries and your remote works. Sometimes you can replace your batteries and the remote doesn't start working again, and that means that the remote maybe have something broken inside. And so your predictions are going to usually follow those if-then statements, and it should, again, be testable, just like your hypothesis. Once you generate your predictions, you'll make an experiment, and then you'll have observations around those experimental trials. And so again, we're looking for falsehood or falsifying those predictions that's tested. And specifically, experiments are carefully planned and manipulated microcosms of the natural world. And you really want to make sure you have controlled experiments. And what I mean by controlled experiments is that everything is controlled except for one variable, and you allow that one variable to change, and then by only changing one variable, you can determine the effect of that variable on a situation. So for example, let's say you wanted to know if applying more nitrogen to the soils made corn grow taller. So you would plant the corn in the same soil, under the same environmental conditions, under the same temperatures. You would apply the same amount of water to each individual plant. The only thing that you would vary is the amount of nitrogen. So one variable that you would change, maybe you give a low dose, a medium dose, and a high dose of nitrogen to the soil. So everything's the same except for one variable, which is nitrogen. And so if you see a difference in the plant height, then you know that it's likely related to the differences in nitrogen. However, if you vary more than one variable, so let's say you vary the amount of water they get and the amount of nitrogen, it's really difficult to tease out which one is contributing to the difference in height. So was it the nitrogen or was it the water that's causing the plants to grow taller or shorter? So that's very difficult if you have two or more variables. So in controlled experiments in scientific process, we really want only one variable to be manipulated and changed. And so once you have that data, you then look at your data, you determine whether or not it falsified your hypothesis. So if the prediction is false, then that means you need to modify your hypothesis, maybe see if something else is happening differently. So for example, with our remote, we put the batteries in it and it doesn't work, then our prediction about the batteries being dead is incorrect. And so we want to look at maybe something is messed up inside the remote and continue our prediction from there. If the prediction is true, then we've really only got at one small part of the hypothesis, and we need to continue testing and refining our ideas so that we can refine and gain a better understanding of that idea and the observations that we are making. And so like I mentioned before, hypotheses can't be proven true. We only can support them or disprove them. So if we've never disproved them, then we've got support for that hypothesis. And so we're assuming it to be correct, right? We're assuming that our hypothesis is accurate because we haven't been able to falsify it. Now here's an example of a controlled experiment where you have a large number of subjects, and you want a large number of subjects because you want to try to encompass the complete amount of variation in a population. If you only have, say, 5 individuals out of a population of 100, you may not encompass all of the variation. So you want to make sure you get that variation within the population. Then you'll randomly divide that population into 2 groups. And you want it to be random so that there's no biases in the data. So that randomization is important. Then you'll randomly assign a treatment to each group. And in this case, we have a drug that should lower blood pressure and then a placebo pill that's not supposed to have any effect. And then those patients will take the pills, the doctors will take measurements, and then they will see if there's a difference between the blood pressure between the experimental group. So the experimental group is the group that received the experimental drug, or the control group, which is the group that received the placebo. If there is a difference, then that will support the hypothesis that this drug lowers blood pressure. If there is no difference, then that drug does not lower blood pressure, and they need to look at developing a new drug and going back into their data to see maybe what causes high levels of blood pressure. So here we have a little video that's kind of going over the scientific method. Make sure my sound. So we'll just watch this real quick.
The scientific method often begins with an observation that raises a question. Next, a possible explanation called a hypothesis is formulated to answer the question. But to be useful, that explanation must be testable. Generally, the hypothesis will lead to a prediction. The prediction is tested by experimentation or further observations. The results are then interpreted to form a conclusion. If the results support the hypothesis, new predictions can be made to further test the hypothesis. If the original hypothesis is not supported, an alternative hypothesis is generated and tested. Here's one example of the scientific method in action. If, while walking through the rainforest, you came across plants which you later identify as lemon ant plants growing alone with no other plants growing around them, you might record and describe that observation. Comparing the density of different plants in the patch to the density of different plants in most other areas of the forest floor, the relative isolation of lemon ant plants strikes you as odd. You collect information such as the area of the patch, the approximate number of plants in it, and the size of the cleared area around each plant. You also look for another patch, make the same measurements, and compare the patches to see whether there's some correlation between the size of the patch and the area of the clearing. Your measurements suggest that the larger the patch, the larger the cleared area. Next, you develop a hypothesis as a possible answer to your question, asking why lemon ant plants are found in otherwise cleared areas. Your first guess is that the lemon ant plants are producing a chemical that inhibits the growth of other plants. To be useful, this hypothesis needs to be testable. Next, you make a prediction based on your hypothesis and test it with a controlled experiment. You might predict, for example, that if your hypothesis is correct, that a chemical extract alone, in the absence of the intact plant, will inhibit the growth of other plants. You therefore select one lemon ant plant and liquefy its tissues in alcohol using a blender to produce an extract of all the chemicals in the plant that are soluble in alcohol. To discover whether those chemicals inhibit the growth of other plants, you prepare two identical plots of ground and seed them both with the same variety of plants. You then spread the plant extract over the experimental plot, but you leave the other plot, called the control plot, untreated. Sure enough, no new plants grow in the area that you treated with the alcohol extract, but plants grow well in the untreated areas. Do these results support your hypothesis? They seem to, but it's possible that the alcohol in the extract is preventing the seeds in the experimental plot from growing. In this experiment, you actually had two variables that were being tested, the alcohol and the extract itself. You need to experiment again, this time with two control groups and one experimental group. In each plot, you add seeds, but you treat one plot with the alcohol extract, you treat another plot with alcohol only, and you leave the third plot untreated. Sure enough, plant growth is retarded only in the area treated with the extract. Plants grow in the plot treated only with alcohol and in the untreated plot. Your results indicate that the chemicals in the plant tissue are inhibiting the growth of other plants. This, in fact, may be your conclusion, that the lemon ant plant produces a chemical that inhibits the growth of other plants. Note, however, that although your results support your hypothesis and by extension your conclusion, they do not prove the hypothesis. For example, perhaps your results could be explained in another way. Perhaps the lemon ants that live on the plant deposit a chemical on its surface that washes off with rain and inhibits plant growth in the surrounding area. The ant-produced chemical might also have gotten into your extract, along with the chemicals produced by the plant itself. A hypothesis can be proven wrong when the results do not support it, but results that are consistent with a hypothesis do not prove that it's correct. Even successful experiments often lead to revised hypotheses and further experimentation. What if the plant extract had not inhibited the growth of other plants? This would mean that your hypothesis needs to be rethought and that you may have to find another explanation. Essentially, you must now look for new variables.
All right, so that finishes up pretty much our discussion on the scientific process, right? So going from observation to hypothesis to prediction to experimentation, generating results, and then gathering a conclusion, right? And then reiterating that cycle as necessary. Now, once a scientist or a researcher goes through their experiments, eventually they'll have enough data and enough conclusions to be able to submit their findings and put it into a story so that we can share that knowledge and research with other scientists in our community and also the public. And scientists publish their papers in what's known as peer-reviewed journals. And with peer-reviewed journals, this basically means that other experts in your field will look at your work, they'll critically go through it, look at all of your methods, look at your reasoning, look at your conclusions, how you did things, why you know, concluded that from your data. So they'll basically go through it with a fine-tooth comb and make sure that your work is sound and that the results and conclusions are appropriate for the work that you did. And so the results in this peer-reviewed work are only valid under conditions that your experiment was completed, right? So you can't take your results and put them in a very different system and expect them to work or come out the same way. You would need to experiment for that other system as well. So it's really important to know and speak to results for the system that you did your experiment in. And usually the most accurate scientific information is in peer-reviewed journals because they go through that vetting process. Now that doesn't mean that there's some research that can make it through that maybe wasn't done correctly or had biases in it or has something wrong with it. But generally, your peer-reviewed journals of scientific work are the primary sources that you want to look for. So they're known as the primary literature. So that's kind of what you want to look at when you look at scientific work. And over time, a hypothesis can actually become a theory. Right? And in order for that to happen, though, the hypothesis has to be broad and it has to have been tested over and over again and not be falsified in any of those tests. And so it has to be supported over the time of those experimental tests. And it also has to explain a broad range of facts, which again goes back to the idea that this theory is very broad and it has a large degree of reliability. So think about the cell theory and the laws of gravity and those types of theories. Those theories are broad. They describe kind of big ideas and they have a lot of support for those theories and the scientific work behind it. Now, what's interesting though is if an experiment comes up and it actually disproves a part of a theory, then that theory can be refuted and revised or even, thrown away in the future. So theories can be modified. And so I want to bring up this idea, or this kind of thing that happens in the public where you'll hear somebody say, "Oh, it's just a theory," right? It doesn't have any value behind it. When actually that statement is as far from correct as it can be. Theories are science most sound kind of bodies of work. Hypotheses are our ideas, and what they really the public really should say is it's only a hypothesis. It's only a guess or an interpretation of an observation. But if something is a theory, then that means it has a large body of work supporting it, and it has been extensively tested over time, and we've not found kind of any evidence refuting that theory. Now, that doesn't mean that it can't be refuted in the future, but as far as we know, based off of the evidence that we have, it is supported by the most sound evidence, right? And so that's a big thing that I want to make sure you take away from at least this, is that, the difference between a hypothesis and a theory, and that theories are not just guesses. They're actually a really sound, what's the word I'm looking for? Tested and supported idea in science. And so the sources where you can get scientific information will vary in terms of how they're written, and then also the quality of that work. We already mentioned peer-reviewed journals. Peer review, again, is the top type of source that you want to use. That's our primary literature. After that, you can go to science magazines and nonfiction books. So like the American Naturalist or National Geographic. So those types of magazines are ones you can look at. So they're usually summarizing peer-reviewed journals and telling you about the work from those peer-reviewed journals. And nonfiction books are generally using, peer-reviewed work, if they're science books, to discuss and talk about that. Now, that doesn't mean that some fiction or other aspects that aren't correct can get put in there by the editors, because a lot of the times the authors of the peer-reviewed journals won't have any kind of say over the final edition of the magazines and the books, so just be aware of that. And then below that are kind of the general interest news magazines or any type of newspaper or news outlet, because again, there's editors, and they're going to interpret and discuss the work in ways that could be incorrect in terms of what the peer-reviewed journal is actually stating. And then lastly, the internet. You really kind of don't want to use websites for any source in science. You really want to stick to peer-reviewed journals and your science magazines and that sort of thing, and kind of leave these bottom two sources out of kind of your research papers and that sort of stuff. But that means I'm not saying you can't use them, because you can be a critical thinker whenever you're looking at this work, right? Even with the peer-reviewed work, you can be a skeptic. You can look at somebody's research and be like, I don't really know about this. I don't really, think these methods were the correct thing to do, or I don't know about this interpretation of their results. That's absolutely fine. And we do that every day in science. We'll, read a peer-reviewed journal, and just because other experts in the field have reviewed that doesn't mean that there aren't some flaws that are found in that research, because no study is perfect. And so you can be a skeptic, and you can critically analyze the work that has been published. So you want to learn how to read graphs. You want to learn what statistics are used in these papers and how to, you know, use those statistics to gain an understanding of what it's actually telling you. So like your chi-square test, your p-values, those types of things, because that really tells you the results in a data form, and you can look at the numbers yourself and see if the interpretations of the authors actually correlate and go with the with what the numbers and the data is saying. And so you want to be able to distinguish between antecedents versus scientific evidence, separate facts from conclusions, because again, the data could say something different from what the authors conclude. So you want to be able to separate those and understand the differences between those. And then understand the difference between correlation and causation. So just because something is correlated doesn't mean that there's a causation behind it. One of my favorite examples to discuss about this is an example where somebody took data from shark attacks and the amount of ice cream eaten, and that data is actually correlated with a positive relationship, which suggests that as you eat more ice cream, there are more shark attacks. So basically, if you eat a lot of ice cream, you have a higher chance of being eaten by or attacked by a shark. Now, you're probably thinking that makes no sense at all. Well, if you think about it, if you go to the beach, generally when you go to the beach, it's hot outside. So what do you want whenever it's hot? You want something cool, you want ice cream, you want popsicles, right? So a lot of people at the beach eat a lot of ice cream. And if a lot of ice cream is being eaten, that means that there are a lot of people in the water. With more people in the water, that means that there are more chances for a shark attack to happen. So even though there's a correlation between the amount of ice cream eaten and the chance of a shark attack, there's no causation between eating more ice cream leading to shark attacks, right? So you just want to be wary of correlation and how it relates to causation. So again, just because something's correlated doesn't mean that that necessarily causes that thing to happen. And so finally, the role of science and society. So with all of this work, all the scientific work, science can improve technology. It can improve the human condition, right? We all have, not all of us, but the average life expectancy of humans has increased due to technology and advances in medicine, which all have roots in the scientific community. But we also need to understand that science has limitations. We only have so much resources, right? We only can go so fast, and how fast we go in terms of generating new ideas and new technology is based off of the current technology that we have. And so we're kind of in this cyclical limitation with that. But what science does let us do is it lets us make informed choices, right? It allows us to make good decisions about policy or how we should handle our environment, or, you know, what kind of things we should do under certain situations. And so science is really, really helpful in those aspects. Okay? And so that's it for chapter 1. And so next you have your module 3, which we will begin looking at chapter 2. Right? And so as always, let me know if you have any questions, if you have any concerns, you can send me an email. And yeah, so that's it. We'll see you in the next module.